Funding for this program is provided by Annenberg CPB to advance excellent teaching.
For over 10 years, at 48 laboratories around the world, hundreds of scientists worked to create one of biology's greatest achievements, the Human Genome Project. It was a cooperative effort that revealed a collection of over 3 billion nucleotides and provided researchers with a genomic database.
The game shifts from the search, rock by rock, pebble by pebble, to the understanding of the whole landscape, and it's a whole new ballgame.
How are researchers using the new tools of genomics as they study diseases like diabetes, as they investigate the mechanism of DNA repair, and as they try to understand the process of viral infection in plants? Genomics is touching every area of study in the world of biology. One of the most basic lessons that has emerged from the study of biology is the unity of all living organisms. Whether looking at microbes in a drop of rainwater, giant trees stretching into the sky, or animals that walk the earth, all organisms are made of cells, and each cell receives its instructions from the underlying genes. But in the last 2 decades, the understanding of biology has gone through enormous change. Researchers have sequenced the human genome and the genomes of dozens of other organisms. Scientists have learned how to genetically modify organisms. And biologists have started to decipher the complex genetic process that directs the development of an organism. Rediscovering Biology looks at some of the most significant changes in 13 different areas, like human evolution, microbial diversity, and proteomics. Using video, web, and print, the series explores new approaches being used to address some of biology's fundamental questions and introduces some of the scientists who are shaping these changes. The series wasn't created for the general public. It was made for teachers who need to understand the new developments in our field. One of the biggest changes in our field is the emergence of genomics, the study of the functions and interactions of all the genes in the genome.
We suddenly can't analyze data with pencil and paper anymore. We have to run computer programs to compare the expression patterns of when genes are turned on and off to say which genes are like each other, or compare their sequences.
Genomics relies on these new tools of bioinformatics to help provide insights as we analyze our risk for disease and as we develop new drugs to treat that disease. But genomics goes beyond healthcare.
It tells us a lot about evolution. It tells us about the unity of all life on this planet. We see the same sets of genes being used and reused across many, many different kinds of organisms. We really begin to understand the tree of evolution in much greater detail by reading whole genomes, which are in effect evolution's lab notebooks, where it's been taking notes for billions of years.
With dozens of organisms now sequenced, researchers are able to use these data to study unique biological processes. One of these crucial processes is DNA repair. Scientists are analyzing the genome of one organism that may help explain how cells work to fix damaged DNA. The organism was first discovered in 1957 when the U.S. Army began experimenting with a new way to keep food safe. Cans of meat were exposed to a dose of gamma radiation, a technique used to kill microbes left in the meat after packing. But sometimes, when the irradiated cans were opened, something was left alive.
What was left was Deinococcus radiodurans. This is a picture of a DNA repair experiment in Deinococcus radiodurans. And the white stuff is the DNA. And the further the DNA is down in this gel, the smaller the pieces are. After a large dose of gamma radiation, the genome is broken up into lots of little pieces, and that's why you get this big smear of DNA. And after 24 hours, The DNA is stitched back together until it's very much like it started. That's the repair of the genome, and that's what we're trying to figure out.
Jonathan Eisen is a researcher at TIGER, the Institute for Genomic Research in Bethesda, Maryland. He believes that understanding how Deinococcus survives exposure to radiation could help humans learn how to cope with radiation and other dangerous mutagens.
It's so good at resisting radiation that it almost certainly has very good DNA repair processes. And that makes it useful not just for studying this one organism, but also for learning about organisms that don't do it as well as Deinococcus.
Researchers at TIGER sequenced the genome for Deinococcus in 2001. Revealing the sequence of all the nucleotides was an important first step. But to truly understand the functions of an organism, researchers need to locate the genes.
So the first thing we do is we take the complete genome sequence, and using a set of bioinformatics tools, we scan the genome to look for where there might be what are called open reading frames.
Starting from a given point, the computer begins reading the sequence in groups of 3, translating them into the amino acid equivalent. One set of 3 letters, A, U, G, can be found at the beginning of a gene. This is a start codon. When the program finds a long sequence between a start codon and a stop codon, found at the end of the gene, this is an open reading frame. But for most researchers, just knowing the list of genes isn't enough.
Just having this list of putative genes doesn't tell us the biology of the organism. We want to know what that gene does, what it does in the organism. Is it involved in repairing the DNA, is one of the things we were interested in. So how do we figure that out?
To determine the functions of an organism's genes, researchers look to large genomic databases containing all publicly known genes.
It used to be that when you were working on a problem, the vast majority of the data that you were studying was data generated on your own lab bench, or at most data generated by a few other people in the field. Now increasingly everyone is depending on large communal data sets that have information that bears on every problem.
When we look for this similarity between our gene and other genes that have been characterized before, we use usually a program called BLAST.
BLAST, the Basic Local Alignment Search Tool, is one of the most common gene matching programs used by researchers.
BLAST basically takes your gene and slides it against every known gene up and down, looking for regions where your gene is similar to those known genes, and lines them up in the regions where they're similar, and then tells you how similar it is to the gene number 1 in the database. How similar is it to gene number 2 in the database, gene number 3, et cetera, et cetera, et cetera, and then it will rank them, giving you the most similar gene as your top hit for your search.
After months of work comparing the Deinococcus genome with all other genomes, Eisen began analyzing the data. But the results were disappointing.
We looked at the complete picture of the entire genome of Deinococcus. Even with a little bit of rose-colored glasses, we couldn't really see that it was different in terms of its likely DNA repair capabilities from anything else that was out there. It had a lot of genes that we predicted to be involved in DNA repair, but so does the bacterium E. coli, so does the yeast, so does human. But those organisms aren't radiation resistant.
When comparing Deinococcus to other organisms, it is clear that they act differently after radiation. After exposure to gamma rays, organisms will sustain numerous double and single strand breaks in the genome. In most cases, the organism unleashes nucleases to destroy these DNA fragments. But in Deinococcus, the microbe might delete or control this nuclease function, which could give the organism time to repair its DNA.
What we've learned, in part, is that we may have been asking the wrong question in the first place. Rather than looking for the genes that make the organism radiation resistant, we might want to also look for the genes in other organisms that suppress their radiation resistance, that are missing in Deinococcus.
But if Deinococcus has some unique system for controlling its nuclease function, that's only part of the answer. The full answer undoubtedly involves a number of genes, both missing and present, and understanding how they work may still be years away. But for Eisen, the study remains compelling.
So we're excited, actually. We're doing follow-up experimental work. We'd like to do more genome analysis with closely related Deinococcus strains. It's a very exciting thing to work on, and it's a mystery still. We don't know how it's able to do this. That makes it more exciting, not less exciting.
Genomics doesn't replace lab work, but it does speed the process of research, allowing biologists to ask new questions. And as researchers continue to discover more gene functions and make that knowledge available to the entire research community, genome searches will become even more accurate and more useful. One area that has benefited from this ever-expanding knowledge base is healthcare. With access to genomic databases, medical researchers have uncovered new tools for diagnosing and treating disease. Scientists now have gene tests for many diseases, including muscular dystrophy and Huntington's disease. Cancer researchers can predict which tumors will metastasize, and drug designers can target specific protein receptors. Genomic tools allow researchers to study small changes in the genome that were previously difficult to find. And for patients with diabetes, a single nucleotide change can have a major effect. David Altshuler is an endocrinologist at Massachusetts General Hospital. For nearly 10 years, he's been treating patients with diabetes. Over 17 million people in the U.S. and more than 150 million people around the world have type 2 diabetes, the most common form.
When I went through my medical training and I discovered that we actually couldn't tell for one patient versus another, why did this one get diabetes and that one didn't? We know what happens. We know the blood sugar goes up, we know the nerves get damaged, the blood vessels, but we don't really know why or how.
In addition to his clinical practice, Altshuler is a senior researcher at MIT's Whitehead Institute for Genomics. Working together with a team of researchers, Altshuler designed a study to test 16 genes that had been previously implicated in diabetes. After thousands of patient tests, the team determined only one of the genes played a role in the disease.
We tested each of the gene variants in each of the patients, and the question was which of them would systematically and reproducibly show a relationship to diabetes.
The main cause of type 2 diabetes isn't inherited gene mutations. Diabetes develops mostly as a result of diet, lifestyle, and environment. But if researchers can discover genes involved in diabetes, they have a much better chance of understanding how the disease works. The gene that Altshuler's team eventually identified is known as PPAR-gamma, a gene present in everyone. But in many patients with type 2 diabetes, the gene has a slight variation, a single nucleotide change. That single letter change can have a major impact. When we compare the genomes from any two people, they are nearly identical. But DNA between any two people differs on average of 1 out of 1,000 bases. These variations in the sequence are called single nucleotide polymorphisms, or SNPs. At the DNA level, we are 99.9% similar. But it is these single-letter variations in our genes that account for most of our distinctive difference from one another. These genetic variations are responsible for the differences in our hair color, our eye color, our physical shape and size. And they can make us more or less likely to get a particular disease.
In theory, what we'd like to do is run along the genome and take all 6 or 7 or 8 million places in the population where there are common sites of variation and simply write down a big table for each variant. How often does it occur in people with disease 1, disease 2, disease 3, disease 4? Take the next variant and in some sense describe all of the relationship between human genetic variation and risk of disease in one big matrix. After all, that matrix contains all the information about how genetic factors play out in risk of disease.
SNPs have helped researchers locate genes involved in several diseases, including Parkinson's disease, cystic fibrosis, and Alzheimer's disease.
There are indeed two SNPs located on chromosome 19 that together determine a reasonable portion of people's risk for Alzheimer's disease. If you happen to have inherited a double dose of a particular SNP spelling, you have a much higher risk, about a 50% risk of Alzheimer's disease versus an alternative.
Bottom line is things seem like they're going extremely.
In addition to helping discover a person's risk for disease, scientists believe that SNP research may help design better medical treatments.
Maybe you'd be better off starting on drug A if you have one set of genetic variants, and better off on drug B if you have a different set of genetic variants. And that could be very useful to identify which drugs will work best in which patients.
Researchers have also discovered that it isn't just individual SNPs that are inherited. Entire sections of closely linked gene variations are passed from generation to generation.
In other words, the variations aren't utterly randomly assorted with each other, but in regions of the genome, they tend to go together in chunks. We call these chunks haplotypes.
Within the human genome, SNPs occur on an average of 1 in 1,000 base pairs. Researchers have shown that groups of SNPs occur in predictable patterns within sections of DNA. These inherited sections of 6 to 8 SNPs are called a haplotype. Within any one section of DNA, it is believed that there are only 3 to 5 different haplotypes throughout the entire population. Scientists are now working on a haplotype mapping project that will help medical researchers quickly compare a patient's genetic patterns with patterns that have been previously identified. That way, they can determine if the patient is at risk for particular diseases. With over 3 billion nucleotides in our genome, finding the 10 million SNPs that may point us towards disease can be a challenge. Haplotypes are like cliff notes. They make it easier to spot the variations. Medical researchers believe SNPs and haplotypes will speed the process of disease diagnosis. And while the search for human disease gets most of the headlines, a large research community is using genomics to understand plant disease too. In a western Oregon cherry orchard, plant pathologist Jay Scheit is searching for signs of one of those diseases. And on one tree, he finds it.
This is the cherry rasp leaf virus. It's a disease caused by a virus that's on this cherry tree. It's characterized by these small enations on the underside of the leaf that looks like the back of a cat's tongue.
The virus enters the tree through a nematode. It's an infection that strikes thousands of trees in the U.S. each year.
Once the tree gets it, there's almost nothing we can do. We have to cut the whole tree out, basically, to control the disease, to keep it from spreading to other trees.
Cherry rasp virus is just one of many viral infections that attack U.S. crops, costing farmers millions of dollars each year. Plant pathologists fight the diseases when they can, but for most diseases, there remain many unanswered questions.
How are viruses recognized as foreign, invasive, unwanted entities that can cause damage and disease? How are they recognized, and how do plants respond to those pathogens?
At Oregon State University's Genomic Research Center, biologist Jim Carrington studies viral infection in one of the world's most studied plants.
The best model organism for studying plants is the mustard weed, Arabidopsis thaliana. And its great advantage over most other plants is it's very little and it grows very fast. If you want to study corn, the best you can do is get 3 growing cycles in a year. Whereas Arabidopsis, you can grow right in your own laboratory, and it grows very rapidly, and you can get a dozen generations in a year, perhaps.
In addition, Arabidopsis has very little non-coding DNA, making it easier to study. Arabidopsis was sequenced in 2000, and subsequent research revealed a list of 26,819 genes. Findings in Arabidopsis research have application in many organisms.
It turns out that the underlying mechanisms of virus replication, virus spread through plants, and then plant responses to the virus, those underlying mechanisms are essentially the same, whether you're talking about Arabidopsis or let's say, a cherry tree. One type of experiment that we do is a microarray experiment where we ask the following question: what are all of the genes in Arabidopsis that are either upregulated or downregulated during the course of a virus infection? We inoculate a set of plants with a virus, and we mock inoculate another group of plants, meaning that they receive no virus. And then we compare the messenger RNA pools in those two plants after some given period of time.
A microarray is a small glass chip containing pieces of DNA called probes or features. A robotic system can spot hundreds of thousands of DNA segments onto the chip. For Carrington's experiment, the chip contains multiple copies of DNA from every known gene in Arabidopsis. Researchers first remove samples from both the uninfected plant and the plant infected with the virus. mRNA is extracted from each of the plant samples. The mRNA is then reverse transcribed into more stable complementary DNA, called cDNA. The infected sample is fluorescently labeled red, while the uninfected sample is labeled green. At this point, in many microarray experiments, the two cDNA samples are combined. The combined sample of fluorescently labeled cDNA is applied to the microarray chip. The cDNA from the sample binds to its complementary sequence of DNA bases on the chip.
We know that the pieces that are fluorescently labeled will stick tightly to the corresponding gene features, but they won't stick tightly anywhere else. So we rinse the chip off after we do the hybridization step, and then we put the chip in a laser scanner to electronically capture the data.
The chip is scanned with a laser, activating the fluorescent dyes in the sampled cDNA. A computer captures this information, calculates the ratio of red and green on each spot, which indicates which genes are expressed in the uninfected and infected plant samples.
The amount of that fluorescence is directly proportional to the amount of messenger RNA in the global pool from Arabidopsis, which is a direct reflection of the gene activity that was going on in Arabidopsis at the time you harvested and took the samples. Yeah, let's look at the scatter plot. This is the microarray experiment in triplicate.
Right.
Results from the microarray experiment are displayed on the computer, which is linked to international Arabidopsis databases. This allows researchers to click on any gene displayed to access all published genetic data.
When we click on a gene, we can learn if there's a known function for that gene. We can learn if the gene looks like other genes for which there's a known function. And depending on how well studied the gene is, it may instantly click to us why the gene is up or down.
Before tools like the microarray, researchers mostly studied organisms one gene at a time. Microarrays and other whole genome approaches have changed the way scientists work.
It's revolutionized the type of information that it gives you, because now we can not only, in an experiment, measure gene activity of genes that we're interested in, but we can also measure the activity of genes that we don't even know to be interested in yet. And the way we know we should be interested in them is how those genes behave during, for example, the course of a virus infection.
When researchers find a gene they believe is involved in plant defense, they can do further investigation through a knockout study, an experiment that studies a plant with a deliberately inactivated gene. At the Salk Institute in San Diego, scientists maintain a large collection of Arabidopsis plants, each with a different gene inactivated. Researchers around the world access the Salk collection for their studies. Scientists can inoculate a knockout plant with a virus to see if the inactivated gene is part of the plant's system for staying healthy. If it is part of the plant's defense, it should be easy to see.
We ought to see a plant become sicker faster, accumulate more virus. It will appear as if the virus is more invasive or virulent on those plants.
Microarrays and knockout studies are just two of the experiments researchers are using to study the genome of an organism. Jim Carrington hopes the studies will help get him closer to his dream of understanding the function of every gene in Arabidopsis.
That's probably our biggest dream right now, but in another few years we may, in fact, answer that to our full satisfaction and be dreaming the next generation of experiments. And that's the really exciting thing about science and using Arabidopsis, is seeing the dream evolve.
The ability to sequence entire genomes and the tools developed for these studies have changed the way we do research, from looking at single genes to comparing entire genomes. But there is still much more to understand.
Genomics has answered one big question: what are the parts? What it hasn't yet answered is how they fit together. It's as if we have the parts list for some vast airplane, but we don't know how to put it together to make an airplane, and we don't understand why it flies.
Even with all the new tools of genomics, the core aspects of biology haven't changed. The basic question of organismal function remains central. Instead, genomics heralds the study of biology on a whole new scale. It offers us a way to simultaneously study the action of many thousands of genes and proteins across many different organisms. Researchers are beginning to catch a glimpse of the whole picture as they strive for a firmer understanding of how our parts list translates into subtle and complex interactions that characterize living systems.
Funding for this program is provided by Annenberg CPB to advance excellent teaching. For information about this and other Annenberg CPB programs, call 1-800-LEARNER and visit us at www.learner.org.